Welcome to Humor. So, whenever a pathogen enters and attacks human beings, it elicits immune response, and this immune response is it can be either cell-mediated and humoral immune response. So, we have already covered the cell-mediated. Now, let's mediated immune response. Let's understand all about humoral immune response. Now, this is initiated by B lymphocytes, which are produced in the bone marrow, and they are processed in pairs patches, or they are also processed in lung associated lymphoid organ, and they stimulate the plasma cells to produce immunoglobulin. Now this is structure of immunoglobulin. It has got two light chains and two heavy chains. Based on the presence of heavy chains, immunoglobulin can be classified as immunoglobulin A with alpha heavy chain, M with mu heavy chain, immunoglobulin G with gamma chain, immunoglobulin E with epsilon chain, and delta chain immunoglobulin is termed as immunoglobulin D. Now both the light chain and the heavy chain have got two regions. One is a variable region where the amino acid sequence varies, and the other one is constant region where the amino acid sequence is in constant. So the variable region of both the light chain and the heavy chain constitute the FAB part of the immunoglobulin, and this is responsible for the pathogen binding or the antigen binding site. Now the constant region of the heavy chain constitute the FC part, and this is responsible for complement binding. Now the light chain is connected to heavy chain with the help of a disulfide bridge, and the two heavy chains are interconnected at the hinge region again by disulfide bridge. Now there is one great importance of this variable region, especially of the light chain. Now this contributes to immunological diversity. So based on limited number of genes, variety of different immunoglobulins can be produced having different light chain because of the somatic DNA recombination taking place with the help of variable light chains. Now coming to mechanism action of immunoglobulin, there are four different mechanisms. So the first is simple agglutination or the neutralization of the pathogen. Second one is by stimulating the classical complement pathway, wherein the FAB portion binds to the pathogen and the FC portion binds to the classical complement pathway. Now this causes activation of the pathway, converting the inactive complement components to the active complement components. For example, C4A 4 gets converted to 4A, which and 2 get converted to 2A. Similarly, there is cascade activation of 3 5, then 6 7 8 9. All these activated complement components, they attack the cell membrane of the host cell, and this results in holes in the cell membrane, causes cell lysis, and this is how it will stimulate phagocytosis. The next one is opsonization. Here the FAB portion role is fixed, it will fix to the pathogen, and the FC portion will fix to the macrophages, and it will facilitate phagocytosis by the macrophage. Next is antibody dependent cell mediated cytolysis, wherein the FC portion, now it will fix to a natural killer cell. Now this natural killer cell will identify the changes in the major histocompatibility complex, and this will help to differentiate normal cell from tumor cell or the virus cell. Now this major histocompatibility complex will the major role of this is to identify self from non-self antigens. So once the non-self antigens in the form of tumor cells or the virus is identified, now this natural killer cells will release cytotoxic granules, and this will destroy selectively the tumor cells and the. So coming to types of immune response, there are two types of immune response: the primary immune response and the secondary immune response. Primary immune response is mainly initiated by immunoglobulin M. Now this immune response appear within 10 days of antigenic stimulation. Now one important feature of this primary immune response is it does not require prior antigenic stimulation. Now the peak is attained by 20 days, and it comes down by 30 days. The activity decreases by 30 days. This is primary immune response. Coming to secondary immune response, this is under the influence of immunoglobulin G. Now this is characterized by a quicker, faster and stronger immune response. Quicker in the sense the Immunoglobulin defense mechanism comes into picture as early as 3 days. Peak is attained faster at 10 days, and it is stronger. It stays in the circulation for a longer period of time. This is about primary and secondary immune response. Coming to immunological diversity means that with the limited number of genes for heavy chain and the light chain, a variety of different immunoglobulins can be produced by this mechanism. Now, this is somatic recombination of DNA. Now, this is the structural gene responsible for transcribing and translating or production of immunoglobulin light chain. It has got variable chain and the joining chain and the constant chain. The variable chain has got 500 segments, joining chain has got 6 segments, and the constant light chain has got 20 segments. So, for example, the first 200 segment from the variable region join with the first 4 segments of the joining region, and first 10 segments of the constant light chain, and this is transcribed and translated to produce new light chain. So in the next step, the first 400 can join, and first 5 segments can join, and first 15 segments can join and can produce different immunoglobulins. So from a single limited number of genes, a variety of different immunoglobulin light chain can be produced by this immunological by somatic decombination of DNA. This is also known as immunological diversity. So this can be asked as a 3-marker. Now, or a reasoning based question can be asked like that. So coming to individual immunoglobulins, immunoglobulin G constitute 80% of the total immunoglobulin. It is called G because it contains gamma heavy chain. Now this immunoglobulin G is responsible for secondary immune response. It means that this immunoglobulin is produced in response to antigenic stimulation. So it can cross placenta and confer confer the antibodies to the growing fetus. It is responsible for opsonization. Opsonization means the FAB portion of IgG binds to pathogen and FC portion binds to macrophage and facilitate phagocytosis of the pathogen by the macrophage. Next, immunoglobulin G also has got this type of mechanism of action where known as antibody dependent cell cytolysis, wherein the FAB portion binds to the pathogen and FC portion binds to the natural killer cell, which releases cytotoxic chemicals to neutralize the virus and the tumor cells. Okay, so next one is Rh isoimmunization. Now, so when this occurs when mother is Rh positive and mother is Rh negative and baby is Rh positive. So during childbirth, what happens is fetal RBC will enter into maternal circulation, and this will produce anti-Rh antibodies in the maternal blood. So in subsequent pregnancy, what happens? These anti-Rh antibodies enter the fetal circulation, and they can induce massive fetal hemolysis. Now, so what happens when mother is Rh negative and baby is Rh positive? First baby is not affected, but subsequent child will be affected with massive hemolysis. In order to prevent this, can be easily prevented by administration of anti-Rh antibody injection to the mother immediately after delivery or within 48 hours after delivery. So this injection is given to in order to provide the subsequent child from developing massive fetal hemolysis. So this was about immunoglobulin G. Coming to immunoglobulin M, it is so named because it has got mu heavy chain and it has got 5 different subunits. They are joined by J chain, and because of the presence of 5 different subunits, this can bind 5 different antigens separately. So this is responsible for primary immune response. Means that IgM is produced without any antigenic stimulation, and all the natural antibodies are IgM in nature. Okay, so and a person with blood group A will have anti-B antibodies. Now, and see, these antibodies are produced without any antigenic stimulation. That means the person having blood group A will have anti-B antibodies, and production of this anti-B antibodies does not require any antigenic stimulation, and they are all IgM in nature, and hence they are called as natural antibodies. Coming to immunoglobulin E, it is so named because it has got epsilon heavy chain, and this is responsible for immediate type of hypersensitivity reaction. Means the hypersensitivity reaction occur within 30 minutes of administration or encounter with the antigen. Now this is also responsible for anaphylaxis. The mechanism is as follows. So when an antigen enters, now this will go and fix to the FAB portion of immunoglobulin E. Now, so this immunoglobulin E, the Fc portion, will fix on mast cell and causes degranulation of the mast cell. Now, this mast cell contains slow reacting substance of anaphylaxis, and it also contains histamine, and these two together bring about massive vasodilatation, bronchoconstriction, and hypotension. So releasing and this reaction is known as hypersensitive reaction, and this reaction occurs within 30 minutes of encounter with the antigen. So the other examples for hypersensitive reaction include anaphylaxis by penicillin injection. So when penicillin injection is given to an individual, so this type of hypersensitive reaction can occur. So that's the reason why we should always administer test dose followed by the total dose to the patient. The other example include hay fever by fungus, and asthma, precipitation of asthma attack by pollen, and urticaria by certain food elements. So some individuals are allergic to certain foods. The moment they consume that particular food, they get to see massive hypersensitive reactions all over the body, and it is manifested as edema, inflammation, redness, pallor, a slight amount of fever. Sometimes it can be localized at the site of entry of the antigen, or it can be generalized urticaria, also hypersensitive reaction. So this was about immunoglobulin E. Coming to immunoglobulin A, also called as secretory immunoglobulin, it is a dimer made up of two units, and the two units are linked by J chain, and it is stabilized by secretory piece. Now this secretory piece provides protection for this immunoglobulin against proteolytic enzymes. Now it contains alpha heavy chain, hence the name immunoglobulin A. Now the main function of this immunoglobulin A is provides protection against all the mucosal surfaces like gastrointestinal tract, nasopharyngeal tract, urogenital tract, tears, saliva, and sweat. So this was all about humoral immunity and different types of immunoglobulins. Stay tuned. The next video will be on multiple myeloma. Happy learning. So please do like, share and subscribe for this video. Thank you.